which is an assistant professor in the math department. And she's going to talk about mathematical models and something that has to do with the effects of COVID. Uduak, it's all yours.
Thank you, Jose, for the opportunity to present. I'm very excited, and I'm going to talk about lung development. Jose talked about COVID. Well, it has a lot of application to what happens with COVID. So I'm not going to go into COVID directly, but at the end of this talk, I'm going to highlight why we need to study lung development and why we have to look right from the embryonic stage to understand how the lung develops for us to understand the diseases that we have in adult lungs. So here we have a human fetal lung. And this is obtained from fluorescence microscopy image of a 9.5-week human fetal lung. If you look at the scale here, we have about 500 microns.
You have to change the slide.
Okay, I did.
Well, I don't see it here. I only see the first one.
Okay, I have to start sharing again. Probably it's not sharing.
No. Oh, now I see it. Yeah.
Okay, great. So here we have a fluorescence microscopy image of a 9.5-week human fetal lung. The scale here is about 500 microns. So at this stage, the lung is going to continue to branch. So it's like a branch structure. The lung is a branch structure, it's a fractal. It's going to continue to branch for many more weeks before we get the fully developed lungs. So the lung starts by branching out from the foregut at about 4 weeks of during development. And then it branches. You have the two buds. So you have the left and the right bud. And then these buds keep branching up to 23 generations. So you have the first generation of branching, and then it continues to about 23 generations. And then you have the fully developed lungs. So why is this important? Clinical data have shown recently that the origin of adult lung diseases like COPD is actually found during embryonic development and in early childhood. By looking at the differences in branching structure, they were able to see how they were able to correlate these differences in branching structure to COPD. So now it becomes very fascinating because some of the molecules like fibroblast growth factor, which is a ligand that is expressed during development, is also found to have effect in inflammation and fibrosis. So we want to understand how does, by the time you understand lung development, you're able to look at how can you treat or understand the linkage with adult lung diseases. Now, most of what we know about embryonic lung development is from embryonic mouse lungs. Like all mammalian lungs develop through the process of branching morphogenesis. So on the left here, we have a volumetric rendering of embryonic lungs, which we obtained from imaging data, confocal imaging data. The scale we have here is about 200 microns, so it shows you what the size is like. In prior research, what I've done have been to look at just a section of the lung, which I highlight in yellow, to look at what happens when you apply forces to the lungs, because we know mechanics affect lung development. I've also looked at fluid to see how does fluid flow, because the lung is filled with fluid during development. How does that affect development? But today I'm going to talk about the new work that we are doing that shows how molecules can be modeled, the expression of molecular factors, how we can use mathematical models to understand how these molecular factors regulate lung development. Based on the work that we've done, we believe that the lung needs these molecular factors. When once you have the molecular factors, it lays out the plan, and then mechanics now influences how it branches out. On the right, we have a segmented embryonic lung that shows the key structures. In green is the epithelial branch structure. So this is now in 3 dimensions where we segment from the image on the left. So we are able to look at these branch structures. The nice thing about this is that we can now use this for numerical simulations. So we have the epithelial branching structures in green. The long mesothelium is also shown, and then I show the mesenchyme. So most of what I'm going to talk about is going to be referring to the epithelial branching structures, which form the future A space. And then we are also going to talk about the long mesothelium and also the mesenchyme. So the mesenchyme surrounds the epithelium. Now, I talked about molecular interactions. So we have lots of ligands, receptors, so many molecules that regulate lung development. The key one I'm going to talk about today is fibroblast growth factor 10. Fibroblast growth factor 10, which is FGF10, is very important for lung development. Lungs that do not express FGF10 do not branch. There's a nice procedure that has been done for decades. It's been demonstrated in pigs, it's been demonstrated in mouse, it's been demonstrated in rats, and it's also used in human in very rare cases to treat underdeveloped lungs, fetal lungs. So what happens is that if you take an embryonic lung or a fetal lung, and then you occlude the trachea, the lung branches with increased branching rate. So it has a 50% success rate in human fetus. So when a human fetus that is not developing at the age of the fetus, probably during ultrasound, this is found out, then they go into the fetus and occlude the trachea. And then after a few hours, it begins to stimulate branching, but it has a 50% success rate and can lead to early abortion. Now, why am I mentioning that? There's an experiment that was done in the mouse, and they included the trachea of a mouse that does not express fibroblast growth factor, and the mouse did not branch. So that implies that fibroblast growth factor is very important for lung branching, and lungs that do not express fibroblast growth factor do not branch. Another important molecule is sonic hedgehog. So sonic hedgehog, which is SHH, is an inhibitor of fibroblast growth factor 10. So what we find is that FGF10 is going to activate the production of sonic hedgehog, and while sonic hedgehog is going to inhibit the production of fibroblast growth factor, there are chains of reactions and molecules in between that regulate this branching. But because of the importance of fibroblast growth factor, which I'm going to show in a few minutes, we focus on understanding how fibroblast growth factor regulates lung development. And in this case, in today's talk, the emphasis is going to be what is the minimum requirement to control or regulate the expression, spatiotemporal expression of fibroblast growth factor 10. Now here is a work by Berluschi, and this was one of the first studies to show the importance of fibroblast growth factor 10. And then if you look here, this is an embryonic mouse at 12.5 day. And if you look here where you have the arrows, shows you the dark expression, shows you the expression of fibroblast growth factor 10. And then if you zoom in into the insert on the left, figure A, we now see that the epithelium bud, which is shown lightly here, where the arrow points in the first insert in figure B, it shows the lung bud, or you call it epithelium branch, growing towards the region of high expression of fibroblast growth factor 10. And it's a nice work, gives a very good description of this process. We talk about elongation, we talk about terminal bifurcation, we talk about lateral budding. They all describe the way the lung branches. When we have a single peak, as shown in C, the epithelium bud is going to grow towards this peak. Observe that the FGF10 is expressed close to the border of the lung mesothelium. That's where you have FGF10 being expressed, and it's a marker for the lung mesothelium because it's expressed close to the lung mesothelium. You have the epithelium bud, it grows towards this single peak. You have an elongation. If you look at the insert in B, where you have a kind of split localization of FGF10, it's believed that with this split localization, the lung will grow, or the bud, epithelium bud will grow towards these different peaks, and this will result in branching. And this is what we call terminal bifurcation. So it grows until at some point you have the splitting, and then it grows towards the different splits, and then it branches. So this is where tissue mechanics comes in, where the mechanics is important for it to be able to branch. But first of all, the pattern has to be laid before you're able to get the mechanics. So for today, we are looking at how are these patterns laid out? How can we model the way the patterns are laid out? We also have what we call lateral budding. So with lateral budding, you have peaks on at the side of the epithelium bud. And what happens is that at this point, you will have regions that are opposite those red regions growing towards those regions, and then you would have what we call lateral budding. The first model by trying to understand lung branching is what we call the viscous fingering model by Lubkin and Murray, and that was in 1995. But I used this figure from a 2015 paper by Murray that gives a very nice description of the viscous fingering model. So the orange and the black layer represent fluids of different viscosity. So when you have a fluid that is a viscous fluid moving into a fluid that is less viscous, then the interface is going to be smooth. But if you have a more viscous fluid, if you have a viscous fluid moving into a more viscous fluid, then you have disturbances at the interface that leads to viscous fingering. And that's what we have here on the bottom. So because epithelium is modeled as a more viscous fluid because of the thickness, because of the density of the cells and how it's closely packed. And the mesenchyme, which is loosely packed with cell, is modeled as a viscous material. And then we now say, if this epithelium is now moving in as it grows in, then it leads to viscous fingering. And we now know that this is not the case, but this helps us think about tissue mechanics. And when we started understanding the molecules that regulate branching, we all looked at this model and said, well, it's just a physical model. But now, because we know that there are molecules that regulate branching, we've now been able to look at other types of models. So this is the model by Lubkin and Murray, where they look at, over here, mu1 is a fluid with a different viscosity, which is the epithelium, and then the mesenchyme is modeled as a fluid with a different viscosity. And then because of that, the results are shown where you have the epithelium and the mesenchyme, you now begin to see the branching, which is caused by viscous fingering. But we know now, because of the seminal work by Meshka et al., that developed an atlas that describes lung development. We know that it starts at embryonic day 12. Well, it starts early on in human lungs. It starts at week 4. However, this is showing the atlas from embryonic day 12 to embryonic day 15. So with embryonic day 12 to 13, it lays out what we call the scaffold of the lungs, and that is colored in blue. And then we have in green that is colored what we call planar bifurcation, those colors are in green. We also have orthogonal bifurcation, and those are colored in red. Now, if we look at these different types of branching, they are very different. Domain branching is similar to lateral branching or lateral budding, where on the sides you have the branches growing out from there. And if you stand at the end and look at the branch, you would see this cross shape. You stand at the distal end and look at the branch tip, you see this cross tip because of the way it's branching. We also have the planar bifurcation, which is shown in green. And then what happens is that the parent branch and the daughter branches branch in the same plane. So in that case, if you look at the tips, you stand at the distal end and look at the tips, you will find that they are arranged in the form that they are arranged, all in a straight line. Orthogonal bifurcation is different. You have a 90 degrees rotation of the boards. So instead of you having it being in the same plane as the parent, it has a 90 degrees rotation. And then you have this, if you look at it from the distal end, then the shape you see is very different, shown here in red. So what this made us to see is that long branching is highly stereotyped. New findings have shown that in humans, when the conducting airways are being formed, that it's highly stereotyped, but outside that you have a lot more stochasticity. Now the question is, what forms these different branching shapes? And what we found out is that the lung surface, the regions where you have red, predominantly is where you have a different structure of the lung. On the edge, we have a different structure of the lung as well. So what we did previously was when we had this reconstruction of the images, which is one of the cool things with when biologists share data and make it available for mathematicians. So when I did this reconstruction, I found out that the lungs where we have orthogonal branching is very different from where we have the planar branching. And I said, could the role of geometry, tissue geometry, play a role in this branching? Now, what we have here in A on the bottom of the image is showing you a slice. Think about a longitudinal slice of this image. You can see the epithelium here shown in green, and that is our region of interest. So we can look at different boards based on their region of interest. And we can also model the loop surface as just a round object surrounding the board with no obstruction. But then we assume that because of the thinness of the edge, it would affect the way this is going to branch out. So based on that, what we did was for us to design a model that allows us to be able to study this branching structure. So what we did here is for us to be able to model the different regions in a three-dimensional model. So we look here, when in this equation, I'll describe the boundary conditions first. So the boundary condition n dot in parentheses d grad C says that there is no flux outside those regions. So why do we say there is no flux? Because those are natural boundary conditions. So those are places that are not part of what we would see in the real lung. So what we say is that we just put it out as a natural boundary condition. There is no flux out there. Now on the top here, we assume that we have a flux K, where we specify a particular amount of flux and said, call it K. So what we are doing, what is different in these two models is we look at a dimension that is fairly similar to what we would find in a real embryonic mouse lung. So we have LE here representing the loop edge, and then LS representing the loop surface. ROI is the region of interest, BA represents the basal surface, EP represents the epithelium, ME is the mesenchyme, MT is the mesothelium. So what we do is that, because this is a three-dimensional surface, I show the top of the board. So here, like I'm going to show with this cup, see if it shows. Yeah, we can have at this top, how is the shape of this top? So here we show the shape is round, and this represents the circular region that we are studying surrounding, which is going to be the top of the region of interest surrounding this long board. We also look at the loop edge. We look at the top to be fairly similar to what we would see in the real lung. And with these dimensions, we now model the diffusion of ligands. We assume that the ligand is produced close to the mesothelium, or we call it around the pleural surface, and then it's going to diffuse in the tissue till it gets to the epithelium boundary. Recall that when we looked at the images previously from the study by Belusky that shows FGF10 expression, we know it's expressed close to the mesothelium, and these molecules shown here in black are going to diffuse down to the epithelium boundary. And in defining this model, we look at a diffusion. We write an equation to describe diffusion of these molecules. We look at constant flux of ligand. So the constant flux is what we model here, where K defines the constant flux. And then what is flux? Flux is the number of molecules per area per time. So the amount of molecules per area per time. Now, when we release the molecules, and in the model, which is in our 3D model shown in D and C, we now look at what happens at the epithelium boundary. We assume that there's 100% binding. In other words, when it gets to the epithelium, it's actually consumed. Might not be the case experimentally, but this is a good assumption that allows us to be able to study the binding rate. Now, we can now measure the ligand flux. So at this point, we now calculate the flux, the number of molecules arriving at different regions of the lung per area per time, which describes the binding rate. Once we did that, we were able to look at lung branching. We looked at lung branching at the loop edge. We looked at lung branching at the loop surface. And what we found here is that a very simple mechanism is capable of directing an epithelium tube to elongate, to bend, to flatten, or bifurcate. And all this is just depending on just geometric ratios of the tissue. This is interesting because we are looking at diffusion of molecules. As we assume these molecules will be diffusing, we know the curvature would actually affect how these molecules would behave. Now, when we are in 3 dimensions, it's very difficult for us to be able to show how the molecules are behaving. So what we do is just look at the top. So the top, I'm using a cup here. So all what I'm showing here is the result at the tip, because we are looking at orthogonal branching and domain branching that branches out from the tip. Here we are not considering lateral branching. We are considering the branching after the whole lung structure has been laid out. So by looking at the simulation results, we look at the top and say, what are the patterns? How are these molecules distributed? So by looking at the distribution of the molecules at the tip of the epithelium, which is in the inner region, we are able to see the first round of simulations is just picking at the top. The first row represents the loop edge, and then we look at different shapes of epithelium. We look at a circular one, and then we say, what if was elliptical or what it was elliptical, and then what are the branching patterns we find. And then we see that the shapes of the branching, which we highlight in pink, are a bit different. And so we also look at the second generation of branching. So with the second generation of branching, we can see for the first one, the branching pattern looks like a C. So if it's growing out at these regions where you have higher concentration, well, higher flux, because this is just mainly the flux we are plotting in blue. And the surrounding region is the mesenchyme, and the outer region, the line circling all these two ports is the boundary, which is the mesothelium. So we can see here you would have just a kind of bending because it's growing out on one side. And then I had someone ask me once, why would it grow differently? Because all of them are getting the same, they're getting FGF10 flux. So why would this make it bend? Now the answer I had, and I always say is this: if I throw $1 coin to you, and then I have someone else stand and throw $100 to you at the same rate, well, at the same, like sending it to you, which way will you go? Are you going to grow towards the $1 coin, or you grow towards the where you get the $100 note? So we believe that you have a bias where, depending on the number of molecules that is getting there, would actually influence the rate of branching. I know that there are cases where if you have excessive molecules, probably you will not be able to see that bias. But we assume that during development that you have this trend where this might not be the case. Now in the second image where we have here, you have a circular board. We find out that we do not have branching as well. You would have it growing and curving out, kind of bending. But in this case, where we have the elliptical sections, and also the boards are arranged in the same orientation as the parent branch, we have branching. And this branching pattern, if we're supposed to start with the parent, and then you have these two different branching dots, and then you have the 4 dot laid out as we would expect when we looked at the planar bifurcation from the work by Mejga et al. Now we also look at the loop surface. So with the loop surface, we now use a different geometry. Now to keep everything the same, so what we have changed is just the shape of the geometry. The volume stays the same. So everything is controlled. So we wouldn't say, oh, it's the volume that is changing. Everything stays the same, it's the same volume. The only thing that we change is just the shape. And what we find is that the only one that gave branching is when the daughter branches were oriented perpendicular, as we found in experiment, to the parent branch. So what this has shown us is that a simple mechanism is capable of generating this planar bifurcation and orthogonal bifurcation, mainly due to geometric ratios. Now, once we found this, well, this is very interesting. And we said to ourselves, well, we have a way, though this model doesn't explain domain branching, which some studies have speculated that it might be due to a different mechanism. But this gave us something for us to move up with. So here we show the domain branching planar and orthogonal and relating to what we showed previously to show that the model is able to capture these different changes. So with modeling, what we are looking at here is that what are the minimum requirements for us to be able to see the observed patterns that we are seeing. And this shows that the geometry plays a key role in controlling or influencing the branching. Now we know that orthogonal and planar bifurcation are completely determined by geometric factors. When it's far away, which I've not shown you the result, when it's far away from the mesothelium, it just grows, it just elongates. That's what we found in our simulations. When it gets close to the mesothelium, then we have a splitting of patterns, as we observe this in simulations as well, and then it grows out into different directions. And then we now see that the aspect ratio describes what direction is going to branch, because the aspect ratio describes of the geometry describes the orientation of the daughter branch to the parent branch. It would be nice if that was all that there was to that, but that is not all there is to that. We know from experimental observations that FGF10 expression changes in space and time. So the question now is, how do we take the models that we've done in the past, those ideas of how geometry affects lung branching, how do we take those models and then expand it into a fully modeled out lungs? Now, when we see here in control, we see that it's a larger size, and then on the right, we have a smaller lung. They're all at the same time point. In other words, that's at the same age. But what we find on the left is that this lung is bigger and has more branching, but the other one on the right is smaller and has less branching. The difference that we see is that the one on the right has been manipulated such a way that FGF10 expression is different in the one on the right. So just changing the expression of FGF10 affects lung branching. And then you have reduced lung branching and growth. So the question is, how do we describe this spatiotemporal expression of FGF10 for us to be able to understand how it actually controls lung branching? We know for the case where we have here where I'm hovering my mouse, we can crop it out and say, okay, it really works with the model that we developed a while, which I showed you a few slides back. And then we can describe FGF10 expression and how it drives lung development. But when you move out the lung, the shape is not like a cylindrical object. So we want to be able to say what affects how the patterns are being expressed. And that's what we've written out models, and we are able to understand how we can use these models to be able to describe lung development. And that's what I'm going to show you next. Now here we have FGF10 expression in the mouse lung, highlighted here in purple. And then we show in A here is the early stage of lung development. ST represents the stomach, and this is a mouse lung. And then the bronchi, that's the arrow, BR, and then TR is the trachea, and the thyroid is shown here, up here. Now, when we go to B, we can see the expression pattern of FGF10, that's embryonic mouse 12.5. The patterning is very different when you have at embryonic day 11.5, the expression pattern for FGF10. We go to embryonic day 13.5, and we see that the patterns are even so much different than what we had previously. And since this is a diffusion process where the molecules are moving around, we decide to use a model that we call reaction diffusion systems to be able to model how these patterns are being expressed. And now the model that we use is, which I'm going to describe a little bit later, it's really been developed in the last few decades, maybe about 2 decades now, or maybe a decade now or more, that shows you how you can extend the reaction diffusion models that we've been looking at in the bulk of a geometry to just the surface or a hypersurface of a geometry. So I'm going to talk about that later. But what we are doing is that we are looking at expression at the mesothelium. So rather than the whole bulk of the lung, we look at just on the boundary, the hypersurface of the lung. And then we say, how are these patterns being expressed at the hypersurface? So here is the mouse lung that we have segmented from microscopic images using simple way. And then after segmenting these lungs, what we do is for us to be able to use the finite element, well, the reaction diffusion models, then we solve it using finite element method to see how these patterns are being expressed. Before I go into the model, I'll give you some notations. So here we are going to describe gamma to be a subset of R3, and then that gamma is a C2 hypersurface. So that's going to define our mesothelium. We also assume that there exists an open state U in R3 and a function D such that this expression is satisfied. Now, based on this, we can now define the unit normal as we have written down here to be the gradient of D divided by its magnitude. Now, once we define the unit normal, we can now write down the tangential gradient of a function, any function described on the hypersurface. So in this case, with the mesothelium, any function defined on the mesothelium, so we think about a function that describes the concentration of FGF10 at the mesothelium. We now could specify that function as W, and then because we are looking at the diffusion and reaction only on the hypersurface, we now can look at just tangential gradient and then looking at how it's changing just on the hypersurface. So because of that, we have a different description of the gradients. Instead of using the normal gradient, we now have the tangential gradient. That's why we have this gamma representing that it's only defined on the hypersurface. So then we have this expression where we consider the first term, the real diffusion, and then an additional term. Then together, this describes the tangential gradient. So once we have the tangential gradient, we can now look into how we describe diffusion. Diffusion process in the bulk of the geometry, we all know how to write down that equation, but when we are looking at just the hypersurface, then we have what we call the Laplace-Beltrami operator. And the Laplace-Beltrami operator is the tangential divergence of the tangential gradient. So since the Laplace-Beltrami operator requires you to take the second derivative of your function W, so you have the first gradient, tangential gradient, and another tangential gradient. So when you take the dot product, you get the Laplace-Beltrami operator. So in this case, what we are doing is that we need the function to be twice differentiable in order for us to be able to write down the expression for the Laplace-Beltrami operator. So here is where we describe the model for us that describes FGF10 expression. So we use what we call the surface reaction diffusion model. And then here is describing FGF10 expression. So the solid case U or bold font U represents a vector where we have U and V. So U represents the concentration of FGF10 and W represents the concentration of FGF10 inhibitors. Inhibitors that we looked at a while ago, which we said sonic hedgehog is one of them. So rather than trying to increase the complexity of the model, we group all of these as inhibitors as an initial model to say, how does these inhibitors, if we are looking at FGF10 expression and inhibition, how does that affect the patterning? So what we are looking at here is that we are looking at UT represents the time derivative. So you're looking at the rate of change in FGF10 concentration as a function of time. And this equals the second term describes the diffusion of because U is bold font, it's not just FGF10, it describes diffusion of FGF10 and also the inhibitor. Uppercase D represents, uppercase D represents the diffusion. So we assume that we have isotropic diffusion. So you have here 1 and then you have D. So if we put this D here, 1 means the diffusion of FGF10, and then D would describe the diffusion of the inhibitors. So F here describes the dynamics. It describes the fact that when you have more FGF10 being expressed, then it's going to increase the expression of sonic hedgehog. And then the more, and also all other inhibitors. And then the more that you have the expression of the increase in production of inhibitors, that would inhibit the production of FGF10. So that dynamics is described using this equation represented in F. Now with reaction diffusion systems, which I'm going to talk about a little bit more in the next slide, we know that we need random perturbations about the steady state. So we know the steady state, which we write U naught for the concentration of FGF10 and W naught for the concentration of inhibitors. We have that A plus B, if we solve for steady state by equating the F equation to 0, then we get A plus B to be steady state for FGF10 and B divided by A plus B squared to be the steady state for the inhibitors, the steady state concentration. So then we have some random perturbations, and then we see how these random perturbations would affect the patterning on the surface of the lungs. So in other words, here we are looking at Turing analysis, not just in the bulk as is normally done. We are now looking at it on the surface of the lungs to see how would diffusion affect these different patterns. Now, with Turing analysis, we are doing this on the hypersurface of a sphere in order for us to be able, first of all, when we started, to be able to check if our results are correct, compare them to known results on the sphere, and then see that the model, validate the model that, verify that the model is developed correctly and the numerics has been done correctly before we go on to looking at the surface of the lungs. So here with Turing analysis, in the absence of diffusion, we know the system is stable based on these constraints on the parameters A and B, which we can solve for by looking at the Jacobian of the matrix. And this is all pretty well-known stuff. So we know in the absence of diffusion, we know the parameter, what we need, the constraints we need to fix. Now we identify the constraints in the surface reaction diffusion system that in the presence of diffusion, the system will be unstable. So what we are looking at with Turing analysis, that you have the system where in the absence of diffusion, the system is stable, but then when you bring in diffusion, then the system becomes unstable. So in that case, we are looking at the interplay, the diffusion, where we are saying that FGF10 is diffusing slowly, and then we have the inhibitor diffusing fast. So that based on these dynamics, we have a situation where we can get patterning. Now the question is, can this patterning represent the kind of patterning we are seeing in the lungs? And if it represents, what are the different parameters that we could change for us to be able to see variations in patterning? So here we define initial conditions. So for us to be able to define these initial conditions, we had to solve this model on a sphere, the hypersurface of a sphere, because it's a lot easier for us to play around with that. So once we got everything validated and these A and B values we choose satisfies the conditions for Turing analysis, we use these parameters as the parameters that we use for the lungs. Now, to solve this, we use the finite element on surfaces. So the finite element on surfaces involves that we discretize the surface of the lung. And when we started with the sphere. So we discretize just the surface into quadrilateral, where all the vertices lie on gamma. And then we describe finite element functions that take the continuous function, for example, of FGF10 continuously defined everywhere on the surface, for example, now in a discretized form. And that makes it easier for us to have a reduced problem that we can solve. So then once we validated a model on the sphere, which I didn't find it necessary to show you the results, but we did validate on the sphere and we're able to compare it with theoretical solutions, we now moved on to the lungs. And we knew our code was correct and everything was fine, so we moved on to the lungs. Now looking at the lungs, here is the mesh. So we are seeing this blue represent the mesh. So we discretize into quadrilaterals on the surface. The lung was reconstructed from simple way, and then we use Blender to be able to convert from triangular mesh that we had on the surface to quadrilateral mesh. And then once we did that, we now ran the simulations on this long mesh. So the simulations we ran was for us to look at what is the effect of varying the lung size. And one thing I didn't mention, which I know I omitted, I have to go back to show you. One thing I omitted here, gamma. I didn't talk about gamma. Gamma is a very important parameter. So gamma here describes the size of the lung. So if you change gamma, it corresponds to you physically changing the geometry of the lung, making it bigger and bigger as you increase the value of gamma. That's what it does. So what we did was to keep gamma fixed initially, and then we used Blender. The initial lung that we started with, I call it 1x. That's the lung we started out with. I call it 1x. And then at that point, we solved the equation, the surface reaction diffusion equation, to look at the patterning. We kept gamma fixed at 160. We didn't change anything except the size. We made it 4 times larger. And then we looked at the patterning of FGF10 on the surface. And then we found that it was different. Now we increased the size of the lung to 16 times. And then we find that the complexity of the pattern increases as we increase the size of the geometry. In other words, the lung size itself, when you look at the reaction of the inhibitors and fibroblast growth factor, what the model is telling us is that as the lung begins to grow in size, the dynamics between the FGF10 and inhibitors will result in you having this complexity in the patterning. So the scale bar that we have here, red represents high expression of FGF10, and blue represents low expression of FGF10. And all what we've done here is to keep gamma to be constant at 160. The only thing that we have changed is to change the size of the geometry. And once we found this, what we went out to do is that we know gamma is a very important parameter instead of just growing the size. So the first result was sufficient, but we decided to look at just gamma, what happens as we vary the parameter gamma, just to get additional information. The same thing here, blue represents low concentration of FGF10, red represents high concentration of FGF10. And then we see that with one time, in other words, the original size of the lung we had, the first row, we have for gamma equals to 10, gamma equals to 17, gamma equals to 160. In other words, we are increasing the size of the lung from left to right. We didn't see sufficient patterning, but we know that the patterning changes with the size of the geometry, where gamma represents size of the geometry. As you increase, gamma corresponds to you increasing the size of the geometry. Now we still went into Blender and changed the size of the lung and make it 4 times larger than what we started with. And then take those lungs back and do simulations using the surface reaction diffusion equation. And then we say, how does the pattern change with 4 times the size? And then we see that with 4 times the size, as we increase the value of gamma, it's kind of like you're over doubling the growth as well, not just increasing it physically, then we increase it with the fact and with the parameter gamma. We begin to see when gamma is 160, we begin to see the patterning begins to change. This still also enforces the fact that when we increase the size of geometry, then we are increasing we find increase in complexity of the patterning. Now, it's still an open question, what makes us have these different complex patterns? But what the model is telling us here is that just the size growing on its own can increase the complexity of the patterning. Now we have 16 times, and then we can see that as we make gamma 160, the expression patterns are very much more complex. And if this corresponds to what we found when we actually, I showed you the images where we showed that initially at the early embryonic stage, the patterns are not well spread out, but as it grows out, then you have the complexity of the patterns begins to change. Now, why is this important? We know FGF10 controls lung development. If, like I showed with the smelly, if you smell FGF10, you change how it's being expressed, then it affects patterning. So this is the first step of the model that we are building to study branching patterns. Now, the cool thing we are going to do next is for us to be able to put those branches inside these lungs. And then we can now look at these different patterns where they are expressed and try to see how they connect to the epithelium stalk and how they are regulating and how they could regulate branching. And this will allow us to test different hypotheses like the diffusion models that we had and all the different models that other groups are producing with different assumptions and for branching. We can test these different models. We can also test the role of mechanics. How does that affect FGF10 patterning? Because we know it does affect patterning. If you apply mechanical force like in tracheal occlusion, it changes the rate of branching. We can now use models like this to test by correlating expression far at the mesothelium and how it diffuses in and interacts with the mesothelium if and when we put in the boards of the epithelium into this geometry and couple them together, we can ask those questions. Now we have a qualitative comparison. Now we see slight correlation in these images. And someone might say, why is it not exactly the same? I didn't expect it to be exactly the same. Someone asked me, why don't you try to run these simulations to get exactly the same patterns? And I said, well, the goal is to show that you have reduced patterning at the early stages when the lung is not very large. And what we want to do next is to be able to add the tubes inside this lung, because right now we don't have the tubes. And then we now ask questions, what can we do to be able to get exactly these patterns that we are getting? And can we grow these lungs from this stage to a bigger lung? And how do the interactions change the patterning as well? So on the left is the simulation. On the right, we show a similarity where at this inset, we know we have a high peak as well. Now here I show also comparing with experimental result where I show the patterning that it begins to get complex as you increase the size of the lung. So here we have 13.5, embryonic 13.5, and then we have more patterning. And if I were to show a larger lung, you would see a more correlation. But we have the striation, the kind of patterns that we are seeing, we see something very closely similar where you have high peaks. So here red is high, so you have high peaks and then you have regions that circles out. So growing these lungs even more, we were able to capture branching patterns, expression patterns that look similar to what we are seeing here in the embryonic lungs. So what we want to do in the future is to be able to incorporate the geometry of epithelium branches into these models. The model we have looked at, in this case, we've not looked at fluid flow. We've not looked at tissue mechanics. We know all these are very important for lung development. So in the future, we are going to incorporate advective transport that carries these molecules, not just diffusion, as the lung fluid is carrying the molecules around, how is this going to affect the patterning? And then when we look at the patterning at the mesothelium and define another model for the mesothelium and the epithelium and couple them together and add fluid and make tissue mechanics, this will allow us to be able to study how all these different factors affect lung development. The predictions of this model lend themselves well for testing in engineered tissue where geometry can be controlled. So that's the beauty of this model. This study was motivated by lung branching because it's well simplified, looks at just the main issues with lung development, and for us to ask what is the minimum requirement for us to be able to get these different branching patterns, the spatiotemporal difference in patterns. And this can also be used to study other branching organs that we have, example, the kidney, which we know that its branches very similar to the lungs, but it uses different molecules as activators and inhibitors to control kidney development. So it's the area of research of looking at branching, not just in the lungs, would benefit from having a system that allows us to couple lots of different mechanisms to see how this affects lung branching. And all this ends up being very important for us to see how can we explain this branching and how can it affect the branching structure and how does it change the branching structure. Then in future, if we know how it changes the branching structure, we can move on to ask how does this affect diseases such as COPD that we know that affect the development. Well, we know it's that you can link it to the developmental point at embryonic stage. You can also link it to early childhood. So this will expand our ability to be able to explore these different avenues. So the work at looking at the spatiotemporal expression of FGF10 patterning was done with my student, Geneva Porter, and I would like to acknowledge support from CISOPEP New Investigator Award. And thank you, everyone.
Well, we're open for questions.
So maybe I can ask the question. A great talk, Uduak. Yeah,
Thanks.
it's nice to see after a long time, actually. Yeah, through this talk. Okay, I just have a quick question about modeling of your reaction term, let's say F term. And how did you actually pick that interaction? Because see, there is a U square and W, so they are not linear, Y square, and also U decreases, there is a minus term, but W does not without, you have a linear term in U and not in W. Is that, this sounds very typical and is specific.
Absolutely. Yes, this is specific. This is what they call the Schackenberg model. It's the F10 because the F10 changes based on the type of model, reaction diffusion model you have. So what we picked was the Schackenberg model, and then we said, because what is going on here is that you have this nonlinear interaction. So it's kind of a trimolecular interaction. So it captures, when you have the plus term, you're describing more production, and then you have the minus term, this is giving you reduction. So this U squared W describes the interaction between inhibitor and, FGF10 and the inhibitor.
So could you have just taken U and U times W and like a square Y?
The reason that why it's squared is that it describes the molecular interaction. So this is a very specific type of molecular reaction that is being described by the Schackenberg model. So that's the assumption that we are building into the model, that so the thing here is that it actually describes how the FGF10 inhibits, I'm sorry, how sonic hedgehog or all other inhibitors inhibits the growth factor FGF10, and also how it also increases the expression of the inhibitors. So this is a very specific model that describes molecular interactions.
Okay. Yeah. Thank you.
Thanks.
Any other questions? Comments?
Hi, my name is Anand Srinivasan. I have a quick question.
Yeah, could you speak louder, please?
Yeah, can you hear me now?
Yes.
So I believe on one of the slides you had indicated that from one of your previous research, advection, including the advection term was important, but I believe in this presentation you have not included the advection term. I'm
That's
just curious as to what your assumptions were for not including advection when your previous research has shown that it is important.
Yes, the reason why we didn't include advection is because we are building an initial model. We weren't even sure if this model will be able to describe the dynamics that we are seeing. So we wouldn't want to start with complicated models, so we always start simple. And then we build up the model. Another thing that makes it easy for us to actually ignore is that the fluid flow goes out. So you think about it pumping out in through the trachea. So you have fluid in the lungs. Let's think about you have fluid in the lungs. Then it's being pumped out through the trachea. So the molecules are being transported in this fluid. But then if you look at the lung surface where the molecules are being expressed, so if you're just looking around that surface, then the effect of the fluid we can assume is negligible compared to inside the bulk of the tissue. So once we start modeling the coupling with the bulk of the tissue, we will actually include the fluid.
Thank you.
Anybody else? Well, thank you, Uduak, for a very, very nice talk. And thank you, everybody, for attending, and have a nice weekend.
Thank you, everyone.